In this video, I'm going to discuss the light-independent reactions. These are also referred to as the dark reactions or the Calvin cycle of photosynthesis. I do want to remind you, before this phase, we had the light-dependent reactions happening in the thylakoid membranes, those stacks of grainer. And during those steps, oxygen was produced, and that is leaving the plant out through its stomata. And then we also had ATP and NADPH produced. These are going to go to the dark reactions to play a role there. So let's look at how the carbon dioxide is fixed here into the cycle. This is the first step that we're going to discuss, the carbon fixation step. We see that we need an enzyme here called Rubisco. If this enzyme is not present, then carbon dioxide cannot be fixed into the cycle. So if it's there, it will fix the carbon dioxide to a molecule called ribulose 1,5-bisphosphate. It is a 5-carbon molecule, and it has a phosphate group attached to each end of it. Therefore, the name ribulose 1,5-bisphosphate. So that is step one, and it will form for us 3 unstable intermediates. These are unstable because they have 2 phosphates there attached to them. This is a 6-carbon molecule, of course, because 1 carbon went in with the RUBP, and now we have a 6-carbon molecule. And on each end, there is a phosphate group attached. So the following step will be our Pgal synthesis. And Pgal synthesis is going to start off by forming for us here some Pga molecules first, and then we're going to see the formation of Pgal. So Pga, these are phosphoglycerate molecules. So the Pga is phosphoglycerate, and these are 3 carbons big, and they still do have 1 phosphate attached to them. So what happened here is the unstable intermediates were cleaved in half. We had 3 of them here at the beginning, and so if you cleave 3 in half, you'll end up with 6 of these phosphoglycerate molecules. Next, what we would need is a molecule of ATP to come in for each of these 6 PGAs to transform them into PGAL. So PGAL is a phosphoglyceraldehyde, and we will see those 6 ATPs come in and leave as 6 ADP and 6 inorganic phosphates. We also will need some of the NADPH to come in here from our light-dependent reactions. Of course, that's where the electrons were picked up, and so the electrons are going to be released here, and an oxidation is occurring there, and it is reducing phosphoglycerate into phosphoglyceraldehyde. So phosphoglyceraldehyde is still a 3-carbon molecule, and it will next, in the next step, exit the cycle. In fact, only 1 of the 6 molecules will exit. 5 will stay behind, and this one that exits will join with another PGAL from a previous turn of the cycle. And if you have 2 3-carbon molecules and, of course, the correct enzyme available here, we see the formation of 1 molecule of glucose. Now, glucose, our C6H12O6 molecule here, can be joined together. If you add 2 of them together, you can form sucrose. And if you keep adding more and more together, you can form starch, and eventually even some other organic compounds can form. And most of the energy will be stored in the form of starch in a plant. So this step 3 that we had here, our Pgal synthesis, is followed by a step here where the Pgal molecules are combined to form glucose, which is then used to form sucrose and eventually starch. This brings us back to what we had. Here happening, of course, was 5 Pgal molecules staying behind in the cycle, and those still are 3 carbons big each, and they will go through a series of reactions next here during a phase we refer to as regeneration of RUBP. So this is a multi-step process, but we just have one arrow here. We're simplifying things a bit over here. But the regeneration step is there for the purpose of regenerating RUBP, some ribulose 1,5-bisphosphate. So for each of these Pgal molecules, we're going to have to bring in some more ATP energy, and we are going to have to bring in the energy to convert with this multi-step process the Pgal or the phosphoglyceraldehyde back into ribulose 1,5-bisphosphate. So as I said, it's multiple steps occurring here. We've simplified it. We don't have all the reactions drawn in there, but we do need 3 ATPs in the end here if we want to produce our 3 molecules of RUBP up here. So that brings us back to the start of the cycle, and if we have more CO2 available, that can also be incorporated and the cycle will continue as long as we have the enzyme, as long as we have carbon dioxide and some ribulose 1,5-bisphosphate. This happens in the stroma of the chloroplast, and of course the light-dependent reactions occurred in the grain where we have the thylakoid membranes.